All right, hello everyone. On my watch, it's 12 o'clock, so we can go ahead and get started. First, let me ask, can people hear me?
Yes, I can hear you.
Right. All right, so welcome to this NURSA Consensome Validation NDSP pre-application webinar. My name is Scott Oxner. I'm the biocuration lead for the NURSA Informatics Group, and with me today is a NURSA Informatics PI, Neil McKenna. The format for today's webinar will be in three parts. First, we'll give an overview of the new transcriptomine feature Consensomes. In the second part, we'll explain the scope of the new NDSP RFA, and finally, we're going to answer questions from the audience. I ask that we don't interrupt the first two parts of the webinar since these are being recorded and made available on our website. Instead, you can ask questions by typing them into the GoToMeeting chat box, and questions will be answered at the end in the order that they're received. So as we see here, the NURSA homepage, for those of you who are not familiar with transcriptomine, this is our search engine for mining publicly archived expression profiling data sets in the field of nuclear receptor signaling. We really don't have a lot of time today to go through all the features in this tool, but I would encourage you, if you haven't already, to read our recent Science Signaling Research Resource article on transcriptomine, which goes into depth on how to query transcriptomine and how to interpret the information in the user interface. And there's a full text link here on the transcriptomine homepage to that article. So first of all, what are consensomes? Let me say up front that consensomes feature is in beta and is unpublished, so we really can't go into too much detail in this webinar, but we do recognize that it's important to give you an overview of the underlying informatics so you at least know what you're looking at. The concept behind consensomes is one that is well established in the field of transcriptomic analysis, and that is overcoming the limitations of individual experiments by borrowing statistical power from looking across multiple experiments. So essentially, it's a large meta-analysis. And what is unique about our approach, we feel, is the biocuration element that informs this meta-analysis. So what we've done is we put all of our curated transcriptomic experiments, or over 200 at this point, into two categories. One, the nuclear receptor that's impacted, and two, the biosample category, i.e. Organ and physiological systems that represent these model systems. So from our science signaling paper, looking at figure 2, what you're seeing here shows these various categories. So for example, an experiment involving rosiglitazone, we would classify in the PPAR pathway experiments. And one involving, let's say, dexamethasone, we would then categorize in glucocorticoid receptor experiments. And by that same token, an experiment done in HepG2 cells would represent the liver in the metabolic category. And MinCap cells, for example, would represent prostate in the male reproductive system, and so on. So to generate a consensome, we take a look at all of the full changes in p-values in a defined group of experiments, and we run these through an algorithm that calculates for each gene the frequency of differential regulation, a geometric mean full change, and a p-value which indicates the probability that the frequency arrived at is due to chance or not. So let's give it an example. So we choose the Consensome tool, and let's say, for example, that I'm interested in PPAR and fatty acid pathway in the liver. So what I do here is from the pathway drop-down, I dial in PPARs and fatty acids. Then in the physiological system category, I pick metabolic, which then prompts us to pick a specific organ, and we're going to select liver. So as you make these selections in this tool, what I would like you to keep an eye on is the counter here at the bottom of this form, which tells you how many experiments map to a specific pathway biosample combo. The software that generates a consensome requires a minimum of 4 data sets. So some pathway biosample categories and species combos won't meet this cutoff and won't have consensome. So be sure to check all these species options. And so in this example, I'm drawing a blank on PPAR, metabolic, liver, and the human. But if I switch species to mouse, now I see that there are a bunch of experiments and a consensome is possible to generate. And so having entered in all of our parameters into the filters, we click Submit. And what we're looking at here is what the consensome looks like. You've got a gene symbol, a discovery rate, which is the frequency that this gene was differentially regulated across the experiments, as I talked about earlier, and you have a geometric mean fold change for that gene, and then you have a consensome score, which is a more user-friendly way of expressing the overall p-value. It's inversely proportional to the p-value. So in other words, the higher the consensome score, the lower the probability that this frequency is due to chance. So to give you an idea of the p-values involved here, a consensome score of 171 is approximately equal to 1 times 10 to the minus 69. Now, in any particular consensome, genes are ranked according to the consensome score in a descending manner. And which this is indicative of the relative strength of the relationship between a gene and a pathway in a given organ and physiological system. So something you'll notice immediately is that we don't provide information on directionality in the consensome. And the reason for this is because of the heterogeneity that goes into the consensome itself. Because you're looking at different experimental designs that involve agonists, antagonists, receptor overexpressions, knockouts, receptor knockdowns, as well as, of course, the strong contextuality of gene expression itself. It makes it very challenging to deconvolute all of that complexity. So what we do is we put direction of regulation aside up front, and we say, you know, here is our best guess as to the relative strength of the connection between this gene and in this pathway in this context. And then we leave it to the investigator to determine whether or not this relationship is positive or negative in their own model system of interest. Now, of course, you know, we recognize that users are going to want to see information on direction of regulation in the underlying data points for their own analyses. So we make this information accessible by providing links to transcriptomize queries for each one of these genes. Take a look at RETSAT, for example. And bear with me. So what we see here is upregulation in red, downregulation in blue, and data points that don't make a standard twofold cutoff here are in gray. So, you know, even though we consider this cutoff arbitrary and disregard it really in our own analyses, we do recognize that it's a widely used cutoff in the community, so we set these data points apart visually. Now, bear in mind that this view is restricted to the pathway and biosample settings dialed in the original consensome. And for our sample, this was PPARs in the liver. Now, if you want to see the regulation of that gene, RETSAT, and other pathways in transcriptamine, you need to go back to the filters, and you can set the pathway to all, the biosample physiological system to all, and the species to all. And now, if you rerun this search, you can see the rest. You can see RETSAT's expression in multiple different pathways. Now, a couple of things I'd like to point out in the consensomes. The first is that each consensome initially is limited to the first 1,000 genes that are available in your web browser. Now, the full list can be accessed by clicking on this download results button, which downloads an Excel file of the full consensome. But a nifty tool here is the search box here. You can filter up to the top 1,000 using this box in the top right corner, which is used for finding multiple genes in a given gene family that are all frequently regulated by the same pathway in the same organ. So, for example, if we enter in ACOT here, you can see that 7 members of the acyl-CoA thioesterase family. And if we enter in SLC, we can see numerous solute transporters that are coordinately regulated by the PPAR family subfamily. So the point here really is if you don't see a gene of interest in the top 1,000, it's a very good idea to go back and download the Excel file to look at the full results. So let's now move on to part 2 of the presentation, the NDSPs. So taking our example here of PPAR gammas in the liver, you can see that the top ranking, within the top rankings, there are a lot of well-characterized members of the PPAR subfamily of receptors. For example, we see RETSAT, and if we look a little farther down, we see PDK4. These are 2 well-known, well-characterized PPAR gamma regulated genes in the liver. Now, this is very encouraging to us since it tells us that we're on the right track with respect to our consensome and ranking algorithm. But what's also clear is that in this and as well as other consensomes is that a lot of these highly ranked genes, which we are postulating are important targets of these pathways, are actually not characterized in the literature. Now, they may have been mentioned in passing in an expression profiling experiment here and there, but it's really only when we look at all these experiments simultaneously within a consensome that we see these genes as frequent and consistent targets of these pathways. So the obvious inference here is that these genes are doing something important in the context of these pathways, which is not yet known. But, you know, as long as the evidence for this is from expression profiling experiments only, it's really still only an inference. And so that's where the NDSPs come in. So what we're seeking are proposals from the community that flesh out the pathway and gene relationship that are suggested by a particular consensome and which have not been previously characterized in the research literature. So here I really need to be clear on what I mean by when we say characterized in the research literature. For example, what we don't mean are a gene that's been picked up on an array study and was casually mentioned in the results or the abstracts. We also don't mean genes that were cherry-picked in an expression profiling experiment and then used for qPCR validation. Rather, what we mean by characterizing the research literature are characterizations using classic hypothesis-driven mechanistic and physiological techniques and assays. So I'd like to give you an example of a gene that has been well characterized in the context of a given pathway. So using our same example, we see here CREB3L3. And let's run the transcriptome query for that gene. And we see, bear with me for a moment, we see numerous data points that point to its regulation in the mouse liver by PPAR, and in this case, predominantly PPAR alpha. And indeed, when we look for CREB3L3 in the literature, we found this paper from 2014 that shows CREB3L3 gene regulation by PPAR alpha, and it's also been supplemented with the observations of a lot of mechanistic and physiological context. So that being the case, a proposal based on the PPAR-CREB3 relationship would be considered less responsive to this NDSP, and that's because it's largely already been worked out. Now, if we take a look at another gene here, for example, PXMP4, we'll run this query and look at its regulation of transcriptamine. Yeah. Again, we see lots of data for PXMP4 being upregulated and downregulated by PPAR gamma in the liver. And when we, however, when we looked in the literature for this gene, really the only thing we saw was this paper from 2009 that was using expression profiling to compare the regulation of PPAR target genes between mouse and human. No real extra physiological context. So this gene, which has not been well characterized in the context, would be considered more responsive to the NDSP. All right, so now if an investigator, of course, has preliminary mechanistic or physiological data which corroborate a previously unpublished relationship between pathway X and gene Y, and the gene is highly ranked in the corresponding consensome, then obviously, their proposal would be strengthened by inclusion of that preliminary data. Now, up to this point, we've been focusing on individual genes and small numbers of genes. We do recognize, however, that some investigators might take a different approach. For example, performing experiments with large genes. Speak, please. Thank you. Again, so up to this point, we've been focusing on individual genes and small numbers of genes. We do recognize, however, that some investigators might take a different approach. For example, performing multiple experiments with large numbers of genes, effectively validating a consensome as a whole rather than individual genes. Anyone who is considering a proposal of this nature is encouraged to contact either Dr. McKenna or Dr. Corinne Silva, the NURSA project scientist, and their email addresses are in the NDSP announcement on the NURSA website. Of course, you can also propose validation of whatever number of genes you want to whatever depth, but of course it all has to obviously be able to be feasible within the 1 year and within the allocated total cost of $75,000. And with the conclusion of that, we'd like to move on to the questions, but before we take any questions, please note that proposals will undergo external peer review. So at this point, we're unable to give any information here on the responsiveness or otherwise of a specific proposal. We would like you to limit your questions to the functionality of the consensome feature. And again, for guidance on responsiveness of a proposal, please email Dr. McKenna or Dr. Corinne Silva, the NURSA project scientist. And as I said earlier, their email addresses are in the NDSP announcement on the NURSA website. They will be more than happy to give you guidance. Okay, are there any questions? I don't see anybody didn't. Is there someone who would like to be the presenter? Can we do that? Yes. line. Lei, are you still on the call? I just made you presenter as requested. So I have a question from Dave Bridges. Is there any programmatic access to Consensome and transcriptomine data, or is it all through the web interface? Yes, Dave, there is an API, and you can email Dr. McKenna via further instructions on how to implement that. Yeah, there's information on the API. If you go to the FAQs, and one of the at the bottom here in the section 4 for transcriptomine, there's a link to the API information. Do we have any other questions? Okay, with that, thank you all for attending, and that concludes the NDSP webinar. Thank you. Thank you.